From the modern synthesis to molecular genetics. What was missing? Chapter 9. In 1868, for example, a Swiss investigator isolated deoxyribonucleic acid, DNA, from a cell's nucleus, but he had no idea of its function. Not until 3/4 of a century later, at the conclusion of some research directly related to the 1918 influenza pandemic, did anyone even speculate, much less demonstrate, that DNA carried genetic information. John M. Barry, in The Great Influenza. The big picture. The modern synthesis of the 1940s combined the theory of natural selection with the expanded understanding of Mendelian genetics. The statistical behavior of alleles in natural populations linked Darwin's concept of speciation to changes in the frequencies of specific alleles. Through the 1950s and 1960s, the abstract concepts of alleles and phenotypes were combined with subsequent discoveries concerning the molecular interactions that translate DNA sequences into specific proteins, ushering in the age of molecular genetics. The discovery that DNA is the genetic material of all living things led to the discovery of its structure. Protein synthesis was shown to take place on ribosomes. Messenger RNA links DNA sequences to amino acid sequences during protein synthesis, and transfer RNA carries individual amino acids to the ribosomes. This led to the discovery of the genetic code, and by 1966, all 64 triplets, codons of the genetic code, were mapped to 20 specific amino acids, as well as 3 stop signals that terminate protein chains. Since proteins carry out most of the diverse functions of cells and organisms, an understanding of the diverse roles of particular proteins in generating biological organization is essential for a modern understanding of evolution. The one-to-one correspondence between sequences of codons and sequences of amino acids explain the various dominance relationships of Mendelian genetics in terms of gene products and biochemistry. Various types of mutations and the phenotypes that they determine could now be understood in terms of diverse roles played by protein sequences in the inner workings of cells.